And okay, I think we can start. I have a few intro words here. So good morning. Hello, everyone, and welcome, fellow coders. This is the first Coders Club meeting where the goal here is to discuss not really experimental results, but kind of the secret sauce that goes into creating the experimental results that you see in digital pathology manuscripts. I'm especially excited to be here today and learn with you from this highly esteemed panel of computational digital pathology experts. Many of them I suspect you already know and use a lot of their code in your own work. A brief explanation of how the meeting will proceed. Each speaker has already submitted 3 slides to quickly introduce themselves, and we'll go over that first. This will include essentially their background, their research interests, and their software and hardware stacks, so you can get some familiarity with the different speakers and their perspectives and where those things are sort of coming from. Afterward, Elias Baumann and Cedric Walker, who are both PhD students at the University of Bern in Zurich, will act as moderators, guiding us through the questions that you've submitted via the Google document provided during registration. If you have additional questions or follow-up questions, please feel free to type them in the Zoom chat box, and they'll do their best to kind of organize them and inject them into the conversation as needed. Note as well that you will be muted throughout the meeting, so the chat box will be the way of speaking with the panelists. Lastly, as well, I think you probably received a message saying that this meeting is being recorded for those of us who are not able to attend today. So I think that's it. And this is a kind of an experimental approach that we're trying for the first time. So hopefully it goes well. And if you have any feedback afterwards, please feel free to email us and let us know what you think. So I think that's basically it. If there's no further questions from our panel, then I think we can jump into the introductions and get the show on the road. I think I will introduce myself first. So my background is in computer science and mathematics. I did my master's degree as well in computer science, in particular in computer vision. Later on, I did a PhD in computational digital pathology many years ago. For a number of years, I was trained as a bioinformatician, so studying kind of the omics side of cancer research, so copy number variation, SNPs, that sort of thing. I'm now an assistant research professor at Case Western Reserve University, a senior research scientist at the Lausanne University Hospital in Switzerland. I'm also secretary of the Swiss Digital Pathology Consortium. If you live in Switzerland or are interested in this, please visit this website here for additional information. Next slide, please. So my research interests, of course, computational digital pathology, in particular, predicting diagnosis, prognosis, and therapy response. I think probably 95% of my work is in H&E images. These are called image-based biomarkers. They computer-aided diagnostics, kind of all of these keywords. Most of my research focuses in the cancer space, but we've expanded out into renal diseases, cardiac space, in particular, looking at transplantation, predicting rejection of transplant patients, that sort of thing, as well as preclinical models with some pharma collaborations that are starting up. And a recent budding interest of mine is really trying to integrate these tools into the clinical space. So we spend a lot of time with IT folks and basically hospital administrators trying to figure out how we can take the tools that we develop and actually get pathologists to use them safely and routinely. I contribute to a number of open source projects. Probably the most interesting one is my blog, which is just my name. You can visit it there. Lots of code there describing hopefully some of the things that we'll discuss today. HistoQC for quality control and Quick Annotator for rapid annotation. Those are free and publicly available. You can download those. Next slide. I have no idea what's on this third slide. Yes, hardware and software stacks. So we have basically 3 tiers of hardware. We do a lot of local development. So each of the students have their own workflow with a GPU. The older ones are 2080s and the newer ones are 3080s. I do most of my development on a ThinkPad P51 laptop, which has a small GPU, but it is a NVIDIA compatible and CUDA compatible. So everything that's built there easily translates to the other environments. We have an intermediate cluster that consists of 4 group level shared, so shared amongst all the students and people that collaborate with us. Each of those servers has 2 2080 GPUs and 32 cores. The main idea there and the reason for that is we do model training there and a lot of experimentation just because we're the system administrators of those machines. So we can rapidly upgrade versions, drivers, whatever needs to be without interfacing with the university in their kind of larger scale IT operation. For output generation, we have a high performance computing cluster at Case Western, and this has, I think, about 60 GPUs of various qualities and sizes connected around. The heterogeneity makes it a little bit tricky to kind of use for, I would say, routine access as well. They're behind a head node, so you have to log in through multiple nodes in order to have access to them. So it's not as viable for kind of interactive debugging and training and that sort of thing, which is what we use the intermediate machines for. Software, I think nowadays it's almost entirely Python based. We have some legacy bits in MATLAB that we're phasing out. I think a lot of it is just stuff that was coded in C that was compiled into MEX for MATLAB, probably some feature descriptors, texture features, that sort of thing. But as we have competent students joining us, we ask them to kind of try and translate over that code so we can maintain a fully Python stack. We use OpenSlide in some form for access to the various whole slide images that we read. So this is either directly or via wrappers, for example, bioformats and some of Lee's work that I hope he introduces. We use 100% PyTorch for deep learning. We never got on the Keras TensorFlow kind of train and focused specifically on PyTorch. We can discuss those reasons if that's of interest. We use lots of Docker and Singularity to maintain dependencies between the different tools and programs and applications that we've developed. Primarily our operating systems, I would say, were Ubuntu. I use Windows 10 and very heavily leverage that WSL2 sublayer. So I think that's it for me. Who's next? Lee.
All right, good to be with you all. So yeah, I'm at Northwestern University here in Chicago. I'm an associate professor of pathology. I'm not a pathologist, I'm a PhD. And I also direct our computational division, and we have a center in the School of Medicine that's focused on machine learning and imaging and signals that I also direct. Next slide. So I have a PhD in electrical and computer engineering. So I kind of studied like signal processing, math and controls, and then combined that with high performance computing, which I was also very interested in. And so I've been working with digital pathology in some form or another since 2005. I became an assistant professor of biomedical engineering at Georgia Tech in 2012 and was there in Emory from 2012 to 2019. And then I moved to Northwestern in 2019. And now I'm here working in our medical school downtown. Next slide. So I think of our research as having sort of 4 components or pillars. And, really the core component is the AI and the machine learning. So it's computer vision, it's developing, new algorithms to, make predictions or to, do fundamental tasks like detection, classification, segmentation, and large images. We also work a lot with omics and omics data because that also comes from the pathology lab. So some of this is, very fundamental machine learning research. It's not just applied to, histology data, but to other images as well. Another big component of our lab is open source software and developing software tools. And I think more and more we focus on that because, these days, I think the impact of a single algorithm is actually, the time frame is pretty short. So you come out with something and then 2 weeks later, someone has something better, right? So I think, developing these open source tools that really help other people do research better is where we focus a lot of our energy these days. And so one of the main tools we have that's a collaboration with my friend David Gutman at Emory is called the Digital Slide Archive. And it's basically an open source ecosystem for managing, annotating, and analyzing digital pathology images. We have a very large and active engagement on GitHub. I think this gets like 15,000 PyPy pulls a month, which is pretty good for this domain. And, we really focus a lot on kind of software engineering and trying to structure things to make them maintainable and extensible. We have a partner called Kitware. Many of you who use CMake probably know about Kitware. They do a lot of scientific computing, and they really just sort of help us with the structuring the software correctly and also the community engagement. And so that's something that we're very excited about. That tool that we've developed has been used to generate over 1 million human annotations so far. Another thing that we do with these tools and also in the algorithm space is to try to produce data sets. So using our platform to engage people all over the world, coming up with creative protocols that allow pathologists to work with people like med students or residents collaboratively to produce large and high quality data sets. And then also utilizing machine learning to improve, the data labeling process, the efficiency and the accuracy of that. And then finally, there's the science, which is, trying to, engage with our clinical collaborators or our basic scientists to apply the tools to really answer questions. And I think that's really where, some of the very exciting stuff happens. And you get to learn a lot about a wide variety of diseases, which is always fun. Next slide. So we have also, as part of our lab, a research scanning operation that's embedded in our clinical histology lab. And so we have scanners there right where the slides are being produced and archived. And so we have a lot of software that we've developed to kind of support that, that pipeline. When it comes to image analysis, we have a lot of servers that we bought that we maintain here in the data center. These are dedicated for our lab, and students, interact with these through SSH and learn all kind of, tricks and tools with port forwarding and things to run Jupyter notebooks remotely. And so that's really an essential skill, I think, is learning how to deal with, server-based systems and to be comfortable with things like Docker. We have systems, with varying vintages of GPUs, which is always a challenge. So we have a very heterogeneous collection here. And, collectively, there's a lot of RAM and NVMe that's part of this. We have Isilon storage, so that's something that's really nice. We never have to worry about storage. Perhaps stupidly, the university agreed to store everything we ever create, and so they have this infinitely expandable NAS system. And so that's really, I think, a nice feature of our lab. So the two organizations where we're really active in terms of software are Pathology Data Science and Digital Slide Archive. And I've just sort of mentioned some of our, highlight repositories here. There's quite a few, but these are the ones that are the most active recently. Next slide. So I will briefly plug that, we are hiring another postdoc. So we have a couple postdocs and a collection of grad students here, and we're looking for another postdoc to join us. So if you like what you hear today and you like this picture of our lab environment, then feel free to contact me. Thank you.
Okay, I think this is me. This is me, unless there's somebody else who has the same profile as me. So my name is Scott Doyle. I am an assistant professor in the University of Buffalo. I'm in the Department of Pathology and Anatomical Sciences, but like Lee, I am not a pathologist or an anatomist. I was trained as a biomedical engineer. I do a lot of teaching. Part of my role here is educational. Our goal is to bring a lot of the tools and science that we develop in biomedical engineering in the AI and machine learning space over to the medical school. And so my job is to teach med students and graduate students in pathology about AI, machine learning, coding, mathematics, and engineering. Before I came here, I worked for a brief period of time at a startup company that was developed out of my PhD called Ibris. And before that, I was a PhD student at Rutgers University in biomedical engineering, where I learned the ropes of computer vision right before deep learning sort of took over the field. So right now I'm sort of between the pathology and the engineering space at UB. Next slide. In terms of research, I'm not going to reiterate. I think a lot of us work in sort of the same domain. So I do a lot of the same things that Andrew and Lee and a bunch of the other folks here are going to do. I primarily work in cancer research as a disease focus, segmentation, classification. My focus tends to be on training strategies. So one of the things that we're interested in are different ways that you could go about training a classifier. So we generally don't develop new algorithms. We are more interested in how you can train the algorithms that we have more effectively and sort of cut down the number of training samples that you need for a particular task. And so we do a lot of human in the loop training, a lot of active learning, and we're studying strategies for federated learning as well to maintain security of the data that we're working with. One thing that may be a little bit different from some of the other folks on the panel is that I also have research projects in quantitative anatomy. We have a very active give donor program here at the University of Buffalo where people donate cadavers to our gross anatomy program for dissection. We actually get around 600 donations per year in the Buffalo area, which is a lot. We supply a lot of the medical schools in the area with cadavers for their gross anatomy programs. And so what we do is we take those cadavers, CT scan them, and then present the data to the students so that they can study the CT scans as they're doing their gross dissection. And so what my lab does is we take that data and we develop tools for segmenting the soft tissue from cadavers, performing biomechanical simulations, and also doing population modeling of statistical shape models, which is nice. And as I mentioned, part of my role in the med school is teaching engineering to medical students and to graduate students in biological sciences. The two big programs that I'm a part of here are the computational cell biology, anatomy, and pathology program, which is what we've called for our graduate program. And I'm also a member of the PhD program for biomedical sciences, which is an umbrella program for biomedical sciences at the med school, where students can do sort of rotations within the departments in the medical school. Next slide. So in the past year, we've been developing a sort of server architecture that we're calling SuniCell, which has a bunch of different pieces to it. It's hosted by our Center for Computational Research, or CCR here. The digital slide archive that Lee's group has developed plays a big role in that. We use that as our interface for some of our projects for multiple annotators to annotate our data sets. So we actually have an R01 project on head and neck cancers and oral cavity, where we have annotators from 7 different pathology sites who are reviewing the results of our AI algorithms, correcting them, and providing training data for the next round of training. And we use the digital slide archive for all of that. We also have an Omero server from the open microscopy environment, which we have hosted there. And then on the back end, we have a bunch of compute power behind to train the algorithms and evaluate them. On the medical side, we also have a microscopy core lab, which has all kinds of microscopy that I never use, like EM and light sheet and all kinds of cool stuff. And we also have an annotation suite, which has a bunch of these sort of Wacom tablets where med students can go in and annotate the samples that we use for our algorithms. As far as programming stack, I think everybody, like most of the folks here, we use Python programming. One thing that we do a little bit differently, again, related to the gross anatomy projects, is that we use a lot of 3D slicer. We've actually used this for pathology projects as well. If we do serial sectioning, we can look at 3D models of tumor, but we use it primarily for analyzing our CT volumes and creating segmentations and models and annotating those. And so, yeah, so that's what we do. Thanks.
Yay, that's me. Yeah, I'll just get a head start. Next slide, please. Yeah, so very briefly about me. I see my slide got mangled up a bit, but that's fine. I have a technical background. I did a master's degree in biomedical image analysis at Eindhoven University of Technology. PhD in medical sciences. So like Scott, I did that mostly during the time that deep learning was not yet there. So after I finished my thesis, it immediately could go into the recycling bin, which was kind of sad, but also good technological progress and all that kind of stuff. After that, I actually moved to the field of pathology. So that was roughly around 2013, 2014. I went to Germany for a bit. Nice country, by the way, should visit if you get the chance. And then I went back to Radboud University Medical Center, became an assistant professor and leading my own research group there. Next slide, please. So what are my main interests? Well, a couple of the things that I think others have already mentioned. I try to be a bit more specific on some topics. So obviously, I have a big interest in machine learning. It's a lot of stuff that we tend to work on. In that domain specifically, we're paying a lot of attention now to end-to-end learning for whole slide images, because we're moving more and more towards problems where annotations are not possible, like predicting patient survival directly from the slides and things like that. I have a couple of recent projects on integrating across modalities, most specifically radiology and pathology. I think those are the most interesting domains because you have the in vivo aspect and the ex vivo aspect, and everything else you can link to that. And actually, we've spent quite some time in our group on software development. I'll get back to that in a bit. Involved in quite some projects. So I think the biggest one is that I'm a work package leader in the Big Picture project, which is like building the European TCGA, but then bigger. So 3 million slides are supposed to be included at the end. So it's a massive undertaking. So I'm very happy to be part of that. PanCAME, another EU project that is on integration of genetics, radiology, and pathology for pancreatic cancer. Yeah, and I won't go into too much detail for the rest. So for people who are interested, you can check my website. I don't have as nice a blog as Andrew, but I still hope for you guys it might be interesting to pay a visit. Next slide, please. Okay, yeah, so on our hardware side, so we also have a heterogeneous cluster, so that seems to be kind of a common theme. So what we run there is anything from a 1080 Ti to fancier. So of course, the new machines we buy have 3080s. We have one DGX A100 and 2 storage servers for intermittent experiment data, which close together are a bit more than 1 petabyte. I think what might be interesting for people who run heterogeneous clusters is to look at the NVIDIA DeepOps framework for installing heterogeneous clusters. So we do that with Slurm. It also has a Kubernetes option. It's pretty well maintained, works with Ansible, and I think it's a good, at least for us, it's a low maintenance way to handle a heterogeneous cluster. So on the software side, more and more PyTorch. So I think that's also quite a common thread. We have historically worked a lot with TensorFlow, so a lot of our projects and students are still familiar with that. Essentially, all of our new students work mostly with PyTorch and associated libraries like PyTorch Lightning, Albuminations for data augmentation, things like that. For our annotations and slide viewing, reading, and writing, we use ASAP, something I developed when I was actually temporarily in Cleveland, briefly sharing some space with Andrew. And yeah, back then for whole slide images, OpenSlide was actually still actively developed. That it's that long ago. And what was not really there were any options for actually writing out slides that contained your results, things like heat maps. There was not really any public library that could do that. So we started from that. Extended it quite significantly. So we actually have a lot of, it's open source. We have a lot of open source extensions, the most recent for reading DICOM whole slide images. And we also have some internal extensions that were in collaborations with manufacturers so that we could read all the different types of slides we were collecting. Something I don't actively work on myself, but as a whole group, we have worked on quite a bit is Grand Challenge. I think that might be quite familiar to many of you. And in that context, we've also run quite some challenges, which I think are known in the community. So we had Chameleon initially and Panda 2 years ago on prostate cancer grading. And Grand Challenge is actually also an open source framework, so you can actually run that locally in your own institution as well. And I think I covered most of the things I wanted to say there, so we'll get into the nitty-gritty later. So let's move on to the next one.
All right, that's me. Good afternoon or evening or morning, everybody, wherever you are. My name is Francesco Ciompi. I've also put these few slides together to give a brief introduction about myself and my research. Next slide, please. Yeah, so I actually studied electrical engineering in Italy many years ago. I got a bachelor and master degree in 2006 in the end. Then I moved to Spain, to Barcelona, because it's a nice place, but also because I wanted to study computer vision and artificial intelligence, and eventually applied to medical imaging. So I did a second master there, and then I did a PhD on machine learning for medical image analysis. I applied back then a lot of, random forest, AdaBoost, subparameter machine models to actually cardiovascular imaging. I did intravascular ultrasound. That was the topic of my PhD thesis. And then after that, because I was looking for a bit of rainy weather, then I came to the Netherlands in 2013 here in Nijmegen, where I did a couple of years of postdoc working on still machine learning or AI, let's say, applied to analysis of CT images for a project on lung cancer screening, working on basically detection of nodules in 3D images, so CT scans. But then after that, I was a bit tired of grayscale images. So in 2016, I switched to pathology, and there it was all H&E and immunohistochemistry and multiplexing. So lots of colors, super nice, which then I decided to stay basically. So I started as postdoc, and then I became assistant professor on computational pathology. And this is still my current position. Next slide, please. Yeah, a bit about my research and the projects I'm leading or co-leading. There is a bit of overlap with Geert because actually we are sharing a lot of things, including the office when we are not at home like today. So my main research focus is on computational pathology. For here I put like 3 main topics. The first one, maybe the most important one for me at the moment, is predictive and prognostic biomarkers in immuno-oncology, with applications mostly on lung, breast, and colorectal cancer, where the goal there is essentially to use AI to try to improve basically the way patients are selected for different types of treatments, as for example, immunotherapy. Nowadays is very popular topic. Another research focuses on kind of autonomous AI, if you want, so automating the diagnosis of mostly screening related biopsies, where there you have a lot of tissue samples that pathologists have to check that are mostly negative because they come from population screening. And there you can think of colorectal cancer, cervical cancer, etc., and other applications. And I'm also started recently to focus more on rare cancers like pancreatic cancer or some form of hereditary gastric cancer also. And some projects I'm leading or co-leading. The big picture project that Geert has mentioned, we are kind of sharing a bit the lead of the work package on computational pathology, on AI. ExoMode is a European project which mostly focuses on the automated diagnosis of biopsies, one of the topics I mentioned earlier. Ignite is a VD grant from the Dutch NWO. I don't have to give you the details, but it's on the prediction of immunotherapy response. And then this Hanard Fonts is the one that focuses on rare cancer. Next slide, please. Yeah, here I want to do a bit of advertisement. Maybe you have heard about this challenge already. It's called Tiger. It's a challenge that runs on the grandchallenge.org platform. It's about tumor-infiltrated lymphocytes in breast cancer, and actually comes after the 2 challenges that Geert has mentioned. So first chameleon, then we went to panda, and then we needed a 3rd animal, so we decided to go for tiger. It's ongoing. It started a bit less than a month ago, and it will run for another couple of months, I think until the end of April. So you can check out the website here, and if you're interested, you can participate in the challenge. And next slide, please. Yeah, this is really has a lot of overlap with what Geert said, because essentially we use the same infrastructure. So he put a lot of details there. I put a figure that I took from the website of the Sol cluster, this is how we call it, and a bit of numbers about how many GPUs and CPUs we have. And actually the software stack is pretty much the same that Geert is also using, because it's what we use in our group with our PhD students. So it's TensorFlow, PyTorch, ASAP for annotations, but also here I call it, yeah, it's called Cirrus Pathology. It's essentially a viewer that you find on the Grand Challenge platform that when, for example, when you run a reader study, that also Geert mentioned, you also have some tools to make annotations. So you could also think of using that web-based platform for making annotations. The Grand Challenge platform that we already mentioned, I want to add that there you can also have some kind of collection of, sometimes we call them apps, so algorithms that you can develop and then put on the platform, and you can run these algorithms on the platform. And essentially you can upload a slide and then pick one of the available algorithms, and then you get the slides processed, and you can download the result. And you can also interact with the platform via a REST API via Python. And the last one is called Host-like Data. It's a very recent software package that we have developed mostly in the context of the Tiger Challenge, because it allows to deal with some kind of hybrid annotation types, when, for example, you have tissue compartments that are annotated, but also cells, and you can have point annotations or polygons, etc. Sometimes it's difficult to find a library that can deal with all these different types of annotations in the same way. So we developed this host-like data that you can find at that link, and it's also linked in the code section of the Tiger website, because we found that it's very handy to deal with the type of annotations that we are releasing with the Tiger challenge. So I think with that, I would like to conclude this part and go to the next speaker. Thanks.
Okay, I think that's me. So I guess you put all the people from the Netherlands in the same block.
Intentional.
I'm Mitko Veta from well, only Geert is actually Dutch. But I'm Mitko Veta. I'm from the medical image analysis group at the Eindhoven University of Technology. It's a group led by Yassine Plaum, and within it there are several assistant professors working on different topics. My topic is mostly focused on computational pathology. Next slide, please. So this is my bio. I graduated from the university in Skopje in Macedonia. Macedonia is like a small country in Southeast Europe on the topic of digital signal processing. Before that, I did a bachelor in electrical engineering. In 2010, I decided to do a PhD, applied to several places, and the first one I got accepted was at the UMC Utrecht on the topic of digital pathology, as we call it at the time. It was still quite a new field in 2010, and it seemed very interesting and exciting. So in 2014, I graduated with a very generic thesis title called Breast Cancer Histopathology Image Analysis, because, well, that's what I mostly did during my PhD. It was almost all in the pre-deep learning era. I did have in my thesis the paper describing the AMIRA challenge, which was at the time won by the group of Jürgen Schmidhubner. It was one of the first applications in digital pathology, so it was right at the end when I actually graduated, when deep learning was starting to become relevant in medical image analysis. In 2014, I moved to Eindhoven. I was already living there, and there was a nice postdoc position, so I decided, well, it kind of makes sense to move to Eindhoven University of Technology. So from 2014 to 2016, I was a postdoc, and then I was promoted to assistant professor, and I'm in this position until this day. I still closely collaborate with the UMC Utrecht Department of Pathology on a number of projects, as we're going to show later. Next slide, please. So my research interests, the application areas are still computational pathology is number one for me. I do have a project in corneal or also in retina image analysis. And I somehow ended up also doing MR spectroscopy, because Eindhoven is the city of Philips and they have interest in that. So we have a nice collaboration on this topic, which was interesting, but also quite new for me. In methodology, of course, deep learning, but lately I'm becoming very interested in causal machine learning. I think it's going to be becoming increasingly relevant in the future, and still learning. And I want to start doing some things with histopathology, but also in some ophthalmology data that we have with causal machine learning. Also, I think that uncertainty estimation of machine learning models is going to become very relevant and interesting, and it's also something that I would like to put more focus on methodology-wise in the future. So from projects, a lot of these are in collaboration with the UMC Utrecht. The premium project is on prediction of response to immunotherapy, so a similar topic as Francesco, but we focus on melanoma. I think your focus is on lung cancer. Also rare cancer types, so we have a project together with the UMC Utrecht on classification of ambiguous melanocytic lesions, and I also have a very small part of the big picture project in the work package that's headed by Geert. The spectrologist project is on generally on deep learning for spectral data, and there I have a small part on MR spectroscopy. I do a lot of education. my contract says 40%, but often it feels like it's 80. I teach 4 courses to in the bachelor program at the university and 2 in our master program. We have a quite large masters in medical image analysis with around 25 to 30 students each year. And I teach and mentor students in this program as well. I try to steer them in the direction of histopathology image analysis. And for example, in this project course I have linked here, that's using the patch chameleon dataset on the Kaggle challenge. And the goal is that the student develops something and submit to Kaggle to basically to evaluate what they have achieved. Next slide, please. So as a group, in terms of hardware resources, we have 4 GPU servers, which 6 to 8 GPUs each. They're mostly RTX 2080s. We don't have any smart way of submitting jobs. What we do is actually quite, because we're not a huge group, we can just easily coordinate who uses which GPUs on which server by talking on our Slack channel. For the research, for research storage, I have switched to using SURF, which is this national computing infrastructure. This was very recently because we had a small disaster when our storage server died before our engineer noticing that one of the disks was gone. So basically we lost a lot of data. So that pushed me to use this national infrastructure, which is quite nice and cheap. They also have some compute resources, and I'm also considering switching to using those resources in the future. From software, traditionally we used Keras. So for my PhD work, I used MATLAB, but at some point in 2014, I was using the Caffe library, if you remember, for deep learning within MATLAB. So that was really a long time ago, 8 years now, I guess. Then we used Keras, and now I have I push people to use PyTorch more for the reasons that were, I think, also just mentioned on the chat. Now there's a nice ecosystem of state-of-the-art methods from the computer vision field that are available mostly in PyTorch. Yeah, we use ASAP and QPath for annotating slides. We collaborate, as I said, a lot with Utrecht for developing the data. And there we have Nicholas Tonicus, who's mostly his job, well, not his job, but he's like often ask him to export large data sets from the PAC system, and we usually annotate them on a desktop workstation using ASAP or QPath, depending on what we actually want to do. I think that's all. Next slide. I think this is the end, basically. Yes. Okay.
Very good. Thanks, everyone. So let's actually get into it. Let's actually get to the talking and panel discussion part. Keep in mind, everyone, that you can still ask questions in chat. So if you see any of the questions and you have a related one that you would like to ask, just put them in chat and then I'll try to add it. And yeah, let's see how this goes. So the first question we're going to start out with is sort of an introductory question. So say you have an interesting idea you want to pursue. What are sort of the first steps you take when you have that new idea and you want to try something out, you want to implement something new? What's the first things that you do?
See if there's actually data.
100% agree. 100% agree. And not only that, not only just seeing if there's data, but I'm also very interested to see the quality of that particular data. So even though, let's say you have clinical data of interest, when you actually physically receive that clinical data, relevant columns may be missing for some of the patients that you're interested in. So you'll lose another 10, 15, 20% of the patients immediately just from that. Maybe their slides are of poor quality, they need to be excluded there. So I think one of our biggest challenges is still getting a kind of pipeline for pulling out good quality data that has ethics approval and all that sort of stuff.
Yeah, well, the other thing that I always do, so a lot of our ideas from it depends on what idea we're referring to. So on the biology side, a lot of our ideas come from our collaborators and the clinicians that we work with, because they understand the pain points of the diagnostic and prognostic process. And that's where we want to focus our machine learning efforts on. When I wrote my first RL1 and I got comments back, one of the comments that really affected my thinking was, we don't have any concerns about the technical feasibility of this project. We're not clear on who's going to care if you are 100% successful. And so one of the first things I do when someone wants to develop a machine learning algorithm to address a patient concern is, if we are successful, what is going to be the change in your day-to-day activities as a pathologist? And that really sort of helps to narrow the focus of the project. And then you can ask the questions about, how are we going to go about doing this? Do we have the data to do it, et cetera?
I think that's a great point, because when we end up reviewing a lot of papers, even if the experiments are scientifically, valid and they're very robust and the classifiers are robust, I wonder how many of them would never be translated into a clinical setting and are more just academic exercises, because maybe the clinicians themselves have a very inexpensive blood test that they'll do instead, that they'll be able to, for example, get the same exact quality of information at 100% accuracy for $1, instead of having to set up a deep learning digital pathology infrastructure to get a 95% accuracy for the same type of thing. So I think having that scope is extremely important as well. As well, one thing I'm interested in and a lot of our work has focused on is looking at quality control of the data. So we tend to see large amounts of batch effects, and that is also going to determine how difficult it's going to be for us to train our classifiers. And if we have, let's say, 5 sites contributing data, and when you look at those slides, each of those 5 sites is completely different in terms of scanner format, scanner microns per pixel, staining intensity, all of these sort of things. This at least kind of triggers in my mind, this problem is going to be harder than it would be if you had a single site that contributed all of the same data. As well, that clinical data is going to be a lot more comparable internally to itself. Right, because as soon as you switch between hospitals, maybe the way that the protocols or the chemotherapies are executed are slightly different, and that becomes another confounder that you have to address. Yeah.
Another thing we tend to do is lit review also, which you're always supposed to do, but usually we're not the first person. I'm not that smart, so I always think somebody else has had this idea before me. So I try to see what else has been done, and a lot of times it is, someone has addressed the problem, but in a very narrow scope, or they did it, 5 or 10 years ago, and so maybe we can improve upon that with more data from more places.
So maybe you saw that I just put like another sort of follow-up question on the slide. So what are your steps regarding quality control? Because somebody also just asked about TCGA. We all know that TCGA can be all over the place in terms of quality. So what's your take there? So what do you do for checking? So you find data, then what? What do you do?
So in our projects, what we do is we have them sort of go over the data that they want us to analyze. We usually use the digital slide archive for this. So we get on a Zoom call and we look at a data set of some randomly selected slides. And I have my engineering students on the call to ask the really sort of basic questions about the data and what we're looking at and what kind of features we're looking for. And it's interesting, that conversation, because clinicians often talk to other doctors. And so they will use terms, or they will assume some level of knowledge that a computer or an AI is not going to have. And my students ask questions as an untrained AI, where they're like, well, what is this blobby thing over here? Or what is this, dark or blue colored thing over here? And they're like, well, that's ink that you put on the thing so that you can orient it anatomically, which as a med student, you already know that. So it kind of goes unstated. But these questions sort of prompt a discussion about, well, the AI is going to have to recognize that, or is going to have to figure out a way to sort of exclude that from the analysis. And so that conversation helps to drive, not just quality control, but also what kind of quality do you need and what kind of noise are we going to have to deal with? Because to a pathologist, that's not noise. That's just what the data looks like. You just sort of mentally exclude that. But to the AI, that we need to be able to identify that and deal with it.
I think there's also a challenge in regards to the scope of that data set. So if you have 10 slides, then you're in a situation where you can actually look at each of those slides deeply. You can manually go and identify regions that you probably want to avoid because they're blurry or request rescans or whatever it is. But as soon as you start looking at 500 slides or 1,000 slides, it's just not really feasible to do that level of quality control for each of them. We built HistoQC specifically for that purpose, right? I tried to manually annotate the TCGA breast cancer cases, and after 2 hours, I just gave up. I was maybe 2% done with that process and realized I had another 500, 600 slides to do. So we've been trying to build more computational tools to at least facilitate that, help us identify outliers that should be removed and that sort of stuff. Recently as well, we've put out a paper that looks at using HistoQC, and it doesn't have to be HistoQC. Of course, I'm going to promote that because it's my own tool. But ideally, you can go and start to extract features from the slides themselves, such as like the color imbalance, the stain intensity, these sort of things, and run basic statistical tests against your output variable of interest, right? So if you're interested in predicting the prognosis of a patient, you should go and see if there's a batch effect that associates all the pink slides are of a good prognosis, all the blue slides are of a bad prognosis. Because if that exists, then that severely limits the way that you can execute an experiment, right? You can't use a deep learning experiment to do that, because the first thing it's going to do is just learn that pink slides are good prognosis, which is obviously rubbish and something that should have been ignored. So I think trying to keep these things into account is consistently challenging.
I'll just add that we should fairly soon have a HistoQC plugin for digital slide archive. So Andrew and I are working on that.
Cool. One other thing I will do with early projects is what they do a lot in regular software development and industry software development, which is iterate as soon as you can. And one of the things that we tend to do is we take a model, train it just a little bit, run it on a large section of data, and let the algorithm tell us what it struggles with and what kind of sources of noise we might get. And sometimes that works really well, where it will right away get sort of the basic classes out of the way. And then we can start with the pathologists looking at the data and the outputs of the algorithm and say, okay, it seems to struggle with the difference between normal mucosal tissue and neoplasm. And so then we have to go in and sort of train it with more examples for that. So that active learning loop is really important in our initial phases of a project.
I think it's worth pointing out that you don't even have to wait until you have, for example, like ground truth annotations to figure out if something has gone astray, right? Because if you run, let's say, a segmentation algorithm and 99% of the pixels in that output image are positive, you don't have to know that that's wrong, right? You kind of have some first order statistical measures that you can also employ just using morphological operations to try and find some of these outliers as well and address them sooner.
Yeah,
I think that incremental approach to annotation is very powerful. There's a lot of projects that are going in that direction. Obviously, it's very hard to get people to annotate things in bulk. And if you sort of take this incremental approach, I think you can save a lot of time and energy. You have to be really careful that you're not overfitting anything though, because if you're sort of cheating and peeking at your test images, yeah, you can really highly optimize those.
Yeah, that's a fair point.
Sweet. Since we were talking about this already anyways, and we have a question for exactly this. So one of the obvious things is that you have to get annotations at some point. And so do you guys have any like efficient ways of getting that apart from just asking a pathologist to do it for you? And any like favorite tools to do it, whatever you wanna share here?
I think I would say, to kick this off, it really depends on like where the people are and how much data you have to share and how hard it is to get the annotations back from those people. So, QPath is obviously a very popular tool. I think that works well if you're like close to the person who's doing the annotation and, you can share the data with them and it's not too big to move around. Our tool allows centralization, which I think is really powerful when you have large data sets or when you're trying to coordinate the efforts of people, all over the world. So I think that sort of local versus that, hosted approach, those are two very different ways to do this.
As maybe a follow-up on that in terms of getting pathologists to do annotations, I think our golden rule is only use pathologists when you need pathologists. So we actually have a small army of medical student annotators, which we just pay. They cost a fraction of a pathologist. And when needed, we have them trained by pathologists. Pathologists check their annotations. But if you need annotations for cell types or clicking nuclei, please don't bother a pathologist unless it's a task where you actually need their expertise.
I think they're most relevant in challenging situations, right? So, cases where you can't rely on somebody with just a basic histology education from medical school, you would definitely need a pathologist.
Yeah, definitely. I mean, if we have to annotate pancreatic cancer, we're not going to ask a medical student to do it. But if you need to have somebody select all the positive cells in IHC slide, generally don't ask a pathologist to do that. They will hate you.
Yeah.
I mean, I think they just
something-
simply won't do it, right?
Or something in between is if you can train somebody that's cheap to do it in one day, then that's also a nice option. That's something we often do with master students. A pathologist can train them for a few hours and then they can do the task, which can then be fixed by a pathologist.
So we try and have a lot of the people, we try and have like the students become competent to at least develop their own annotations. I'll say with like 85%, 90% competency, so that they can do a lot of that bulk work and then send to a pathologist for final review, where a pathologist can say, yeah, I agree with this. And the benefit of that is that once the student's actually able to do it, it speaks to Geert's point. We're not wasting the pathologist's time to go and click on these individual cells or whatever it is. But then they start to have better technical ideas of how to use available technology to address the problem because they're more experienced in that domain space. And I also try and suggest them to email the pathologist with an email, right? And we discuss this ahead of time with our pathologist, so it's not rude, I would say. But the subject is, is this a positive region or whatever the question is, and then it's just a single screenshot. And the pathologist can receive that on their phone, and maybe they're in between meetings or whatever it is. They can quickly look at it and will receive like just the letter yes or the letter Y or the letter N. So at least there's kind of a more frequent interaction as opposed to doing all of this stuff blindly and then dropping it on them and hoping that it was right.
See, we do. So one thing about efficiency is to reiterate, we let the classifier run as soon as we get, a small batch of training samples, and then we use that to drive the discussion to make things more efficient. The annotators themselves are trained by our clinical collaborators. So they have residents and fellows and med students that they train. And then we have a sort of weekly meeting where we go through the results of both the AI and the manual labels on the digital slide archive instance that we have. And so in terms of tooling, I can't speak highly enough of Lee's work because we use it. We've been using it for the past year, and it has really transformed the way that we do this kind of labeling process, but also the way that we have conversations about the labels. Because one thing that we experienced is that, labeling for segmentation for pathology is not how you learn this stuff in med school. You don't circle lymphocytic infiltrate on an exam. Like, that's not something you can do. And so you get these situations, and my colleague has said to me, she's like, I end up doubting myself more than I ever have as a function of this project, because we're being asked to do things at a level of specificity that we've never been asked to do before, because the classifiers are operating on sort of like micron level scale. So part of that uncertainty or ambiguity about definitions of classes comes into our development of the classifier. And that's why I advocate for one of the first conversations you need to have is with your doctors, and sit them down and talk to them about this. And continue to talk to them and have these in-depth discussions, as you get version 1, version 2 of your classifier running, so that you could talk about how to efficiently annotate.
And one thing to add maybe is something that we are using sometimes when possible to guide people who are making annotations is to, for example, if you have to annotate on H&E and you have access to the tissue, we often do immunohistochemistry to target the specific type of annotations we have to make. So say you have to annotate a tumor in H&E, and you can cut another slide and do cytokeratin staining, or if you have to annotate mitosis, you can do PHS3 immunohistochemistry or CD3 for T cells and stuff like that. So that that helps also, say, guiding and checking the type of annotations you make, even sometimes with little supervision from pathologists, basically, because then when you have like kind of a better reference standard or ground truth, basically from immunohistochemistry, it's just easier. So then typically the students that also Geert mentioned earlier, we can train them initially about the topic, and then show them also the reference in immunohistochemistry, and then it's much easier also to do that. Also for some complex projects. The thing is, for that, you need to have access to the tissue, and you need to be able to do the immunohistochemical staining.
Mhm.
And you need collaborators that are able to facilitate that, and it's non-trivial, I think, for a lot of people. For a number of years, when we first started out, we didn't have access to those sort of things, and I think it's only now that people are seeing the power of those approaches and the power of digital pathology in general, do they feel like it is a good investment of their time and resources to help us build these types of data sets.
A lot of them need publications for their promotion. And so I think this is increasingly, no, seriously, this is increasingly becoming easier to do than like wet bench science or, other things they might be doing, staring at, thousands of cases and writing reports. So, I think, you can engage people on that sort of basis as well.
Yeah, we do that for our med students. So part of our CCBAP, the computational cell biology, anatomy and pathology program, the students are learning these algorithms in their classes alongside when they're learning pathology. And so part of their coursework may involve, annotating things on QPath or with the DSA, or contributing to some of these algorithms. And then the med students, there's a big push. So this is maybe a conversation if you're at a place where you have access to medical expertise, either in students or in faculty, to have an institutional incentive to work with your computational pathologists to develop these data sets and develop these projects, because it shouldn't be the case where we are basically asking them for annotations for our own benefit. Like, they should be able to be incentivized to help with these projects, because it helps, like Lee said, it helps their careers as well.
I find that, when we start with people, the first time you ask them to do something, it's they're very reluctant, but after they get used to using the interface, it really gets smooth. And you ask them, and there's much lower activation energy required to get started. But you really have to, I think, be prepared and know what you're doing and not kind of fumble around. You have to have your protocol ready and have everything completely set up and documented, and not just kind of like learn as you go.
Yeah, I absolutely think so. I mean, I think especially the pathologists we work with are super busy, and they know implicitly when their time is being wasted, or when they start to get frustrated with something. And I don't think it's fair to them. And I think it's really our responsibility to create a easy kind of workflow for them. And I think that's one of the benefits, maybe I can pitch all of us. That's one of the benefits, I think, of collaborating with people like us. Even if you're interested in running your own digital pathology experiment at your own institute, just, you could you could certainly contact me. I think everyone else on this panel is pretty friendly as well, and ask for advice on how to set things up, or how to at least approach a problem, so that you can start off aiming in a good direction. If you don't do that, I would at least suggest to start using small data sets. So don't go and say, here's 1,000 images. Let's, can you go and annotate these? Start with a small region of interest, and then make sure that at that stage you're already getting the type of annotation and the quality of annotation back that you're expecting, because it's very possible. I mean, keep in mind that this is translational science, so there's really two completely different groups of people and their understandings of science in the world there. And if you just say, annotate these cells, I have no idea what you're going to get back. If you're not very specific and you haven't had that conversation, you can get back something that's really not useful. And that's, I think, our fault for doing that, right? It's not the pathologist's fault. It's our fault because we've asked them to do it and not provided a good background for it.
I'm going to disagree with Andrew. My comment is, I think that if you tell someone, I want you to annotate, let's say, tumor because that's what I ask people to do, right? And I want you to annotate, just this type of tumor. If you get back an annotation that you didn't expect, that's helpful information. That's like an interesting thing that happens, right? If I tell someone, can you annotate lymphocytes? And they come back with a really detailed annotation, or somebody else comes back with a really broad, like circle around the area, those are different ways that that instruction is interpreted by the annotators. And that's useful information, because that speaks to what we talk about a lot. And I don't think there's been enough research on this, is, how the class definitions that we have are too simplistic and can be interpreted in different ways. Because you have hard annotations, you have soft annotations with hard boundaries and soft boundaries. You have ontological definitions of like, this is a cell, this is where the boundary of the cell is. So you have like the right annotation versus a fuzzy annotation or a weak annotation. And so that's all part of the conversation. And I would hesitate to say that an annotation was done wrong. I think instead it's an annotation instruction may have been interpreted by someone differently. So the language that you use to describe this, I think, becomes part of the pipeline and part of the experiment.
A constraint though, right? I mean, I don't think you want to have like thousands of disparate annotations. Maybe it's like an interesting one-off approach.
But if you do, right, that's useful information, because that means that your framing of the problem was based on an incomplete understanding. Mm-hmm. Right. It means the way that you had that conversation and the way that you gave those instructions was based on maybe your own understanding of what a tumor constitutes. Right. Mm-hmm. So we have this whole thing about, is central necrosis part of the tumor, or is that something else? Or, cracks in the tissue, should that be included in this polygon or not? Those kinds of conversations, I think, are really useful.
All right, so in the interest of time, I think we got to move on. The next sort of thing is, so say you have your annotations, you have your data ready. How do you preprocess your data? And the question also particularly asks about, how do you store it efficiently? So how do you get it fast to your GPUs? How do you get it fast to your models, whatever? So how to store data in a good way?
Yeah, so I can maybe kick this off. So we actually collect most of the data we use ourselves from our archives. So lots of manual labor, cleaning the glass slides, making sure there's no dust, scanning them. Then we generally anonymize them, and we convert them to a standard TIFF format, because that's still like the most widely used version. We would consider switching to DICOM at a certain point, once that's mature enough, so that ecosystem is really starting up a bit now. In terms of pre-processing for experiments, so what we would generally do is, for first initial experiments, just to see what kind of numbers you can get, is directly train on the slides, which means that you just extract patches on the fly using multiprocessing, etc., with your favorite data augmentation library. And then once you want to get really into the hyperparameter optimization, at least, so I have most experience with TensorFlow, but I'm 100% sure that PyTorch has a similar pipeline. Store the stuff in the format that your deep learning library likes. So for TensorFlow, that means sharding it using the TensorFlow data sub-library framework. Then it's very easy to profile, see where your bottlenecks are, make sure that you saturate your IO, saturate your CPU, and that your GPUs can do most of the work. And if you then want to compare 20 architectures, efficient net to B1 till B9, that goes super fast, and your data pipeline will not be the problem. I don't think this question is on normalization and stuff like that, so I will not mention that, but that can, of course, be part of your pipeline.
I'm going to put in a follow-up soon, but first go on this.
So I think we take a slightly different approach in that most of the time, looking at Geert's papers, I think that we have a lot less slides than Geert does. So we have to be a little bit more targeted in how we approach these sort of things. I think what we'll typically do is extract patches, not on the fly, but since we have limited data, it's reasonable to pull out the areas of interest, and we'll put them in some type of high-speed backend storage. So lately I've been using PyTables, which isn't the best, and I'm thinking about moving away from that. But the benefit there is that you end up having a contiguous byte stream that's stored on the disk that can be sequentially read or randomly read very, very quickly. But as well, by pushing all those regions of interest into a single file, I use a program called VM-Touch, which is available in Linux, which basically will push that from the hard disk into memory. So assuming that you can create a single file that contains all of your data, like an H5 table, something like that. If you could create a single file or a series of files, it doesn't have to be a single one, that can fit into your machine's memory. You can use something like VM-Touch, which will push that entirely into that memory, and you'll never touch the hard disk again, right? It'll never have to load data from the hard disk. And as a result, even if you have a terrible old not solid state, but one of the old platter drives, you won't experience any efficiency decreases there, because you'll end up loading that data at the maximum speed, since it's sequential reads and the hard drives have look ahead and all this sort of stuff. One thing I would be extremely careful about doing, that some folks have done in the past, is to try and save individual patches as individual images. And one of the reasons for that is you end up paying a very high storage cost for that. First of all, in just trying to access the directory, searching through all the inodes of those particular files. But as well, the challenge is that, let's say you have a 32 by 32 by 3 patch. The minimum size that hard disks nowadays will save is 4 kilobytes, right? That's just the inbuilt block size of that hard disk. So any file that's less than that size, or that doesn't line up on that boundary, you're paying for the rest of that space. So in theory, you can have a very small data set that, let's say, would take 1 gigabyte, but it ends up taking up the equivalent of 10 gigabytes on that disk, and you still have to pay that 10 gigabyte loading cost when you access it. So you end up having an order of magnitude less performance by working with smaller individual files. So there's really value in kind of putting all of that stuff together to optimize efficiency.
I would say be very careful about intermediate formats, right? So, when things get stored in an SVS or NDPI, JPEG compression has been applied. If you want to generate some intermediate representation, tiles or something like that, if you apply lossy compression again, you can really introduce artifacts, right? And so you have an option of either paying that cost or storing it in a lossless format where you're going to really, take a single slide and blow it up to like 30 gigabytes, right? So, just be careful how compression is applied. If you're transcoding, that works okay, but it's also very difficult to do.
We try to minimize the number of steps of storage between the raw data and what we're using for analysis, except where it's going to make it much more efficient. We try to sort of have it where you have code that will generate your patches, or will generate your pull the annotations from whatever your platform is, and that code is sort of fixed. And then if we need to regenerate stuff, we'll regenerate it, apply the transforms or apply the normalization steps to the data computationally. And then if we need to, you can delete those sort of intermediate steps and keep the code. Now, if you're doing like a big project where you have a lot of data, then it's more efficient to store it and compress it the way that Andrew was talking about. I don't know if this is the right place to talk about it, but one of the issues that we run into is the sort of simplicity, but sort of more brute force and inefficient approach versus a more complex, but more efficient approach. As someone who has to deal with a lot of students who don't have a background in computer architecture or programming, it's just conceptually easier for them to understand that they have a bunch of images in 2 different folders, and they can tell PyTorch, here's an image folder, the classes are subfolders, now train. There's an overhead associated with that computationally, but there's also overhead for the students to sort of then figure out like, okay, what is PyTables? What are HD5 files? I've never heard of that before, right? And so you often, not just for data storage, but also for every step of the pipeline, you have to make those trade-offs in terms of like getting results quickly versus getting results most optimally.
I agree with Scott. It's a case of efficiency versus ease of use. I mean, in my opinion, streaming formats like TF record have been much faster, even compared to H5, which I was using before. But well, when you get to Torch and you just want to load the images, it's much easier to implement, much faster implementation. But yeah, again, efficiency versus ease of use. That's what you're looking here.
As sort of like a follow-up question that goes into the same direction, like if I mean, you also have annotations, probably something like that. How do you store those? Do you use like a JSON? Do you store them as masks? Depending on what do you do, I guess, as well. But what do you do there?
We use JSON for everything for the most part. Any kind of text-based file format will work. So XML is something that ImageScope uses, Apereo ImageScope, and a couple other tools will use XML. We use JSON because that's what the digital slide archive uses. And then we have Python scripts that if we need a mask, it'll generate a mask from the JSON file, and sort of all the code for figuring out differences in image sizes and sort of calculating from a so essentially you get a polygon of coordinates, and then from that polygon we will scale it if we want to operate on different magnifications. We will either use the histomics functions, or we'll write our own functions to provide that sort of back and forth. But we store everything in text for the most part.
I think most of the stuff we're trying to transition to GeoJSON, which is a specific kind of flavor of JSON, which is well understood by many libraries across many different languages, and it's a standard language, and that allows for even better compression. There's a version of that called, I think, WTB, which is WKB, well-known binary, which is a binary equivalent to GeoJSON, which is even more densely packed. So if you have to store large amounts of it, I think that's a very reasonable way. And because it's plain text, it's very easy to parse, even if you don't have a JSON parser, which works perfectly well on it. It's fairly easy to parse. My only concern, I think, is it becomes a question of how many annotations there are. So if you're annotating millions of cells, I probably would just save a binary mask, just because it's going to be easier to like visually look and compare against. But if you have something that's very large, like a single tumor region, probably a GeoJSON is going to be an easier format to kind of move around and keep sane. And I don't think there's going to be any long-term issue with saving binary images either.
I think the serialized formats that we use on Digital Slide Archive, they do have limitations, like you said, when we're going into like, many hundreds of thousands or millions of cells on a slide. And we're working to improve that. The issue is not really the storage space on the disk, but like, if somebody's viewing that image, they have to stream all that data down from the server to the client. So, we're going to try to create some new, binary methods to improve that.
I'm gonna ask a question that I probably should know the answer to, which is, can the annotations on the Digital Slide Archive, can they accommodate holes in polygons?
Yes, they can. It's not currently turned on in the version that's posted right now, but that's a blocker issue. So it's enabled, in the deep, bowels of the software, but it hasn't bubbled up to, the user interface part yet.
Got it.
We have been going around that issue. So what we did was, for those holes, we had another annotation inside that particular annotation just to fill in those holes.
Yeah, that's what we tend to do as well.
Yeah, I'm sorry about that. We will get that fixed, sooner than later.
That is, by the way, a question from my annotators. I said, I'm going to be talking to Lee later, so I'll ask him.
Well, I think this illustrates
publicly
point.
in front of hundreds of people.
Yeah.
I mean, this illustrates a point that like Kitware, they're amazing software engineers, but like they don't know anything about how these people look at these images. It's up to us to tell them that. And so you're going from the pathologist to somebody like me to Kitware.
Yeah.
And, there's a lot that gets sort of lost in between. So,
let me tell you a story. I'm on a project right now for one of the surgical teams at Mount Sinai, and they have these 3D models that they're creating, and they are using like the built-in Microsoft model viewer to annotate them. So it's basically like MS Paint, but for 3D models. their biggest complaint, and they contacted me because they wanted to build better software to do this analysis. Their biggest complaint was that if they write text to annotate a part of the slide and they rotate, the text rotates. And so it's reversed. And they were like, if you can fix this, we'll pay you whatever you want to build a new piece of software. So that's like where their concerns are coming from. So I just thought that was funny.
That is funny.
Sort of, oh, sorry. Go ahead.
Sorry, on the storage, I mean, there's a discussion going on in the chat about converting the SVS to TIFF. What we are looking for now is the ZAR format. There's been a recent paper on OME and GFF. That's what we are looking for. And we have just tested a few examples. We have compared that with DICOM, SVS reading, and TIFF as well. And ZAR is at least 3 times faster than, in our tests, 3 times faster than DICOM and 5 times faster than SVS in reading any random regions. And that's what we are going to use for the whole slide images. If anyone is interested, there's open source code available for that as well.
So if anyone knows anyone that,
yeah, so I think it's very important to get people to consolidate around a format. You know, nobody loses by us not using another format. Well, maybe somebody does, but you know, I think it really, it's very important. It's one of the most critical things. It's a really boring issue, but like it causes so many problems.
It's a boring issue that literally every one of these discussions ends up at, which is formats are annoying to deal with. So let's all just pick a format. And I think I agree with the comment that someone made earlier that it seems to be coalescing in the DICOM direction, but that's kind of a conceptual coalescence. It doesn't seem to be translating to the actual data sets themselves. Like,
yeah. I mean, it's also that, you know, DICOM is a standard, and so you can have many implementations of that standard. And so I think that's actually not a bad thing. If there's a standard first and then, you know, people can compete on the implementation, you know, that's fine.
Yeah.
There's some new scanners that are now producing DICOM natively, and that's hopefully gonna.
Yeah, what do people use to convert their slides to? Does anyone have software that they use to convert to DICOM? The only one that I found is put out by Orthanc, which is like a DICOM viewer for radiology. They have like a DICOMizer in C++.
There is from Sectra. I think Geert might. I think yeah. So Geert has just put the link there as well. So yeah, there's for DICOM conversion.
Okay, I'll take a look at that. Yeah, we have open. We have Olympus slides, VSI slides, and those we've had a heck of a time getting them to work with other software. I think they work with QPath, but they don't work natively with the digital slide archive, so we have to convert them. But I'll see if this works for our slides.
So Scott, I requested documentation about the VSI format from Olympus, and they sent me by FedEx a giant envelope that was extremely heavy of just a printed out documentation, right? It's like, and I'm started reading, I'm just like, this is hopeless. This is so complex. Yeah. So I feel your pain on that.
They couldn't have emailed you a PDF.
Yeah, I don't know.
That seems environmentally unsound.
Yeah, it was one of the more bizarre moments. So.
So one of the follow-up questions here as well was that one of the recent hypes is like doing GAN normalization, GAN stain normalization, virtual staining, that sort of thing. What are your thoughts on that, in particular relating to like loss of signal because the GAN converts your stuff to something else that's not there, something like that?
So I have 2 thoughts that I'd like to put out there and see what others think. First, I find that training GANs to do this can be tricky, and you end up like training multiple models, and oftentimes you'll have like crazy artifacts. You'll be like, oh, this was a bad model. This was a bad model. This was a bad model. Oh, this one converged to something that's reasonable, and then kind of use that kind of process of elimination. So I find that frustrating. I don't know if someone has a better approach or code that consistently converges to something that's fantastic and artifact free, but I would be very open to using that going forward. My second concern is in regards to understanding what the impact of using that is on the data itself. So I can imagine, and we've put out a paper that's under review now, but I can imagine that you start to impart biases from the training data onto that testing data. And one of the concerns is that if you're using, let's say, handcrafted features and you want to extract nuclear texture, am I 100% guaranteed that applying that GAN normalization is not going to negatively affect that texture feature? I don't have that assurance. And in fact, I think it's actually the opposite. I think it is really likely to go in there and tinker with it to the point that I can't be confident that what I'm getting out is real signal. So I have hesitations there. I think there's some use cases where it may make sense, but in the stuff that we do, I'm still pretty hesitant and on the fence about it.
Yeah, I am a huge curmudgeon about GAN normalization.
Sorry, what? What was that?
Curmudgeon. It's
curmudgeon.
It's what happens when you become over 35.
I'm trying to figure out how to spell that. Hold on. Oh, it's with a C. Yeah. Okay.
Yeah, I mean, maybe I just haven't seen the evidence, but if you are, for example, concerned about conversion, lossy conversion of data from your original scan to JPEG, there's no justification, in my opinion, for using GANs, which introduce all kinds of unknown and very difficult to understand artifacts into your training set. I mean, the way to deal, in my opinion, the way to deal with normalization is about looking at data from as many sources as you can and grabbing as much actual data as you can. Now, if you're limited in the amount of data that you have, then you can do augmentation. GAN for augmentation, maybe, but I would like to see, I actually have a student now who's working on a project where he's comparing GAN augmentation versus using actual samples, which introduce actual real world variability into your training set, which I think is far more valuable. If you don't have the option, is it better? We'll find out. We'll do the experiment and we'll find out. But with regards to, like, this is, we did an experiment a couple of years ago with stain normalization, not GAN based, but just regular stain normalization, and found that stain normalization does not eliminate batch effects. The only thing that eliminates it is training on data from different places. So getting more variability in your data set, not just variations on the same data set, at least for pathology, is really important. And I think it's tough to get around that. The GANs are not the solution to that.
So as an author of a paper doing GAN normalization, I can respond a bit to that.
Excellent.
So in a positive way, yeah, I completely agree with you. The best way to make your network robust to variations in slides and batch effects is adding more data from diverse sources. So that, I think, maybe there's somebody in the call who disagrees with that, but I hope not. Thoughts on GAN normalization? Yeah, so I think, so one thing comparing them to old normalization methods is, of course, we also did some OR normalization methods, like comparing it to things like Machenko and stuff like that. So that only modifies color information, and that's just one source of variation, of course. So there's also texture variation. Some scanners tend to sharpen images, some don't. So that's an aspect that traditional methods cannot fix, and generative adversarial models theoretically can. I do think that with GANs, artifacts can be a serious issue. We decided to try something different. So what we did in our paper is actually doing a residual GAN. So actually the GAN doesn't predict an entirely new image, but it predicts the difference between the original image and the image it should look like. And that, for a large part, eliminated unrealistic artifacts, like that it would hallucinate tissue in the background that was not there. So that helped quite a bit. What we did do is test it on top of data augmentation. So we started an experiment just without data augmentation, and then look at generalization. That's, of course, horrible. Then if you add data augmentation, it generalizes a bit better. But the normalization even added performance on top of that relative to the data augmentation. For the experiment that was mentioned, I'm quite confident that if you add real world data, it will always be better than adding GAN generated data or GAN normalized data. So in that sense, I don't think there's a discussion. So I think GAN normalization can work. Virtual staining, I am also a curmudgeon about. I think it's completely impossible to, for example, generate a CD8 stain out of a H&E stain, because there's no way you can get from morphology which are the cytotoxic T cells and which are the memory T cells, for example. I don't think that's possible. So that's a completely different discussion. But on the normalization front, I wouldn't say I'm a fan, but I think it has its use.
Just stain the tissue, right? I mean, it's not that hard. So
Agreed.
Yeah, we did it. I had a master's student do an experiment where we went from IF staining to H&E staining, which was produced, good results, but from a practical perspective, like you say, if you've already got the IF stains, like just do an H&E stain.
And it's a bit like virtual staining from like PAS to H&E or H&E to PAS. I think that might actually work, but like immunohistochemical stains, I think you have to be very careful if you're not making things up that if the information is actually there in the morphology.
So the GAN thing is interesting. There's a paper that I really like from Stanford that looks at irrelevant style transfer. Maybe people are familiar with this, but they took histopathology and they applied style transfer from like classical paintings and things as augmentation during training. And I think during training you have a lot of freedom to just make the images look however you want, and if it improves performance, that's good, right? So, it depends, I think, on whether you're trying to, augment data for training or whether you're trying to like really standardize images during inference and, produce the best results there.
I think that's a great point. For me, I would be very concerned about seeing an external held out data set that undergoes significant pre-processing before being exposed to inference and then claiming that model is robust. I would look at that very skeptically.
Well, the real test is you have data from 3 different sites. You train on data from one site and test on the second. You train from data from 1 and 3 and test on the second. And then you train on data from 1 that's been GAN augmented to the same data set size as 1 + 3, and then test that on 2. And then you see what the difference in results are. Because what I've seen, and maybe this was just a bad experience, but I've seen papers, abstracts, presentations where they talk about using GAN to generate an infinite number of training sets, which is not possible. That's not a real thing. So my big thing is GANs are constrained by the variation that they're exposed to when you're training the GAN. You're not going to be able to create variability out of that sample size that's going to add to the kind of biological variability that you might see in terms of the expression. You could get around some of the computer vision stuff like differences in staining, differences in contrast, right? That's fine. But I'm skeptical that if you have, for example, a tumor grade that you're trying to run inference on that is not present in your training set, I don't think that the addition of GAN is going to help recognize that structure. And I'm 100% open to being wrong about that, but I would need to see the experiment.
All right, then let's move on to something a bit related, but sort of also on hype topics. Like transformers have sort of like dominated ImageNet and all that sort of like challenges, like especially like the image transformers, the vision transformers. But recently there's again been the rise of like ConvNet papers that have been put out. There's like the popular ConvNext paper, but also the EfficientNet v2 is like working super well. So what's your thoughts on transformers in digital pathology, also considering exactly like those kind of publications?
Yeah,
I would say, sorry, go ahead, Geert.
Oh yeah, can do it very briefly. So in general, for like the next generation of convolutional neural network architectures, I think you just need to run a couple in a cross validation on your training set, because for one problem, like ResNet 301 might be the best one again. And generally, if you use state-of-the-art architectures, you're talking about percentage points or maybe tens of percentage point improvement, right? So it's not the main issue. Which architecture you use, the data and how you evaluate and how you train it is much more important than the architecture. At least that's my opinion. For the transformers, yeah, I think there might be some use there. So I wouldn't immediately do all your patch-based systems with transformers. I don't immediately see the use there. But I could imagine that for people who are know the CLAM framework, right? Which does sort of weekly supervised learning with attention based mechanisms integrated with multiple instance learning. I think you could make a similar model with transformers where you can maintain like spatial awareness of where the different areas originate from, which CLAM does not. I think there might be some use case there, but it's not something we have really explored. So I wouldn't use it like just replace your ConvNet with a transformer. Not like super confident that that would be very sensible to work on, but I think there might be use cases in like weekly supervised learning where you have some use of attention mechanisms.
I think you need a good rationale to do it, and that is a very good rationale, is, you know, preserving the spatial relationships. You know, we all look to the sort of general vision community for inspiration, but I mean, one thing I would say is our data is very different from theirs. You know, they benchmark on things like ImageNet and COCO, and you know, in a lot of ways, our data is much more simple. It's sort of made from more primitive elements, blobs and textures and patterns. So, you know, just because something, you know, shatters a benchmark on ImageNet, you know, I don't think you can expect that, you know, it's going to have a similar impact on our problems and our performance. So that would be my comment.
I would also argue that our data is more complex than those computer vision ones, simply because the data that we have is much more noisy. So if I'm looking at like pixel level annotations and ask you to circle a cell 50 times, you're going to have different boundaries on that. You can have differences, and you know, Geert probably knows this quite well in terms of Gleason grades, right? So at least when ImageNet says that it's a picture of a dog, it's definitely a picture of a dog. And I think that exposes you to a whole new class of algorithms that allow you to do hard negative mining without worrying about injecting additional noise. Where a lot of the stuff that we have, and maybe you guys have better quality data than I do, but a lot of the stuff that we have, if you naively do hard negative mining and just start injecting this stuff, sometimes you end up with worse classifier performance because you're just boosting the noise instead of the actual signal.
I would say it's not even noise. It's just, you know, these structures appear in different conformations within the boundaries of an image. And like, if you're doing face detection, like most faces look similar to one another. You can't say the same thing about tumor from a computer vision standpoint. And to that end, I was actually having a conversation the other day and saying, like, you know, a lot of computer vision focuses on faces. But a lot of it is also on sort of morphotypical faces. I haven't seen a lot of research or a lot of outcomes where you're dealing with faces that have, you know, non-standard features or appearances. We've come a long way in terms of skin color and hair and things like that. But you know, I think a lot of these challenges and a lot of these benchmark data sets are very highly curated, very highly tailored to specific problems. And like Lee said, you know, smashing a record on ImageNet could mean going from, you know, 97 to 97.3% performance, which is great. You know, there's a branch of engineering that's focused on that. But in our world, it's like, okay, so am I still going to give this patient chemotherapy? Probably. Like, it doesn't change the outcome, right? Going back to the question of impact and talking to your pathologists, you know, if you bump your DICE coefficient of segmentation up by 0.01, you know, those maps may look the same. It may not influence what happens to that patient.
Perfect segment into the next like sort of follow-up question. Yeah, when do you consider like performance improvements meaningful? So you just said a 1% accuracy increase is not going to mean anything to you. So maybe the others can also chime in, like what's relevant for you when you look at performance evaluation as well.
So
I think yeah.
Let me clarify just one thing. 1% is what I was just referring to was segmentation performance, right? I think it's very different what you're actually classifying, right? Because getting 1% of patients more accurate, that's a big deal. Like that actually is significant. Getting, you know, if you show someone two different segmentation maps and they look basically the same, quantitatively they could be different, maybe even statistically significantly so, but that's a different thing from saying, you know, all of a sudden I'm classifying 1% of my patients more accurately. That's everything, right? That's what you should be focused on.
So I think some of the points that people have brought up in regards to, let's say, like training students and people becoming familiar with these things, there's a cost in changing your technology stack, right? And there's a cost in trying out different models, and there's a cost in having people try and learn how to, take a transformer and start working with it and what those results look like, what the training patterns, right? So this is all a cost. So I think the real root of this question is, what is a reasonable evidence that you could see in another field that says it's worthwhile for me to pay this cost to try and obtain better performance? I think there's two caveats here. One is in ImageNet, it's a heavily data mined approach right now, and it's probably overfit, right? You see like 0.02% improvements. And I think a lot of that is specific to that version of ImageNet. There was a paper that came out where they created ImageNet 2 using the same exact curation approach that they use for ImageNet 1. So they basically said, oh, we've searched Google for pictures of dogs and we included 15 pictures of dogs, just like the original ImageNet. So it's literally a one-to-one matching of ImageNet. And they tried the latest versions that were the highest performing, and the performance dropped like 15-20%, which indicates to me that there was some generalizability issue when we were overfitting that specific component. The other thing that troubles me is that I know for a fact that if you randomly train at a deep learning network 10 times, you're going to get 10 different models, and you're going to get 10 different results. And the standard deviation of those results is important to understand as well. So anytime I see a paper that says, oh, we did a single training and we have a 5% improvement, like that could be minus 5% if you randomly trained it again from random weights to begin with. So nowadays, I'm also looking for a kind of standard deviation in there and say, well, we're always within kind of this band. And that is additional evidence that would make me feel more confident. But for me nowadays, if it's not at least a 5% improvement, I'll read the paper, but I probably won't pay with the technology. And that's in absolute performance, not in relative performance.
I think it's like a traditional statistical power thing. So, 1%, I would say, well, what's this data set look like, right? If you have a massive data set that covers all these different, histologic variants and things, then 1% could be really meaningful. But, if it's just a small, noisy data set, then, it probably doesn't matter.
Yeah, a point from the chat is that, it also depends on the metric that you're talking about, right? Accuracy is kind of a generic but not very useful performance metric if you're more concerned about positive predictive value or sensitivity or specificity. I think one thing that should get talked about a little bit more is the idea of cost, risk, and performance as being three different concepts that we often treat the same, but the reality is that risk is associated with making the right decision if it's a probabilistic classifier. And we should probably start talking about that in a little bit more nuance when we talk about performance, like what exactly are we referring to, and what are we classifying more accurately or precise or specifically?
Okay,
so to answer the question, 5%.
Perfect.
Yeah.
Let's move on, since we don't have that much time anymore. Say you did all your trainings, you do whatever you did, and then you want to look at your results. How do you keep track of all the different experiments you run, and how do you also store your results? Because you've probably got to look at them again and again, right?
So I do a lot of yelling at my students to give me more graphs and to show me historical graphs. From a practical perspective, we write code to do all of this. So your reporting of your experiments and your reporting of your runs should all be done in the sort of code way. What I don't like to say, and I tell my students this, is do not store results in, like, do not create graphs using Excel, for example, because that's usually their first go-to, because you do have to reconfigure the way that you display results as your experiments may change over time, right? So as the criteria that you're using or as the metrics that you're using change over time, you go from, I want to get an algorithm that just works, to I need to have an algorithm that subdivides these classes and provides different performance metrics for each class, or produces a confusion matrix. I need to see the train validation curves. I need to see examples of the maps of the outputs. All of these things sort of come about as the project develops. And so I tell my students to save everything and have scripts that will generate outputs and artifacts that we can use to examine during our research meetings to see, are you improving the model? Are you getting better results, and so on?
I think one way could be
tools that frame. Sorry, go ahead, Sean.
Yeah, I mean, just wanted to say one way could be to use tools like Git, use version control. Another way could be, I mean, sometimes I use that is just keep a copy of, at the top of my script, I have a script which just makes a copy of my code in a particular format, just all the code with the results, which helps me get back to those to that code which produced that result. But I think version control Git is much better way of doing it.
We use Git a lot, but I think it's not great for keeping track of like parameter settings and things. And I do a lot of kind of what Sean is mentioning, which is, commenting different, hyperparameter settings and having that evolve sort of at the top of my script. I'm very intrigued by these sort of platforms, and people are sort of pasting some of them in the comment section, that, it's a framework that allows you to sort of run your experiments and track and link the metrics to the hyperparameters. But every time we've kind of sat down and looked at those, there's kind of a high startup cost. Like you have to sort of integrate your tools into that framework. And so, I'd be really interested to know if there are tools like that that are easy to use.
There's some that are coming out now, which are like Python. I don't use any of those. I'm really terrible with this. So I was waiting to see what other people said. But I do this very poorly. But there's some now where you basically import a Python library and you can point it at a server that you set up with Docker, expose a port. So it's a very easy setup. And I can't remember the name of it right now, otherwise I'd say it. And you can basically go and use replace print statements with like, push this to the server. And then each time you start the script, it'll create a new kind of output log of that script. And you can save metrics, you can save images as well. You can kind of save all that stuff. And I agree with you. The startup cost used to be massive. I think it's finally starting to get better. But I would note, you still have to be super disciplined in order to do it well. Because if you leave that comment at the top of your script that says, hey, try number 1_done_version 2_version final, regardless of how you organize the rest of that script, whenever you look at those comments, they're still going to be meaningless to you.
Yeah, people are talking about weights and biases, or WANDB, whatever, however you say it in out loud. I've looked at it. I think that the startup cost thing is one thing, but really the discipline that you just mentioned is really important as well, because what'll happen is, in a perfect world, yes, this is kind of how you would keep track of your experiments, but very rarely do we have like, okay, this is a model and now we're doing hyperparameter tuning. So we're going to run the same thing with different hyperparameters and see what we get. These tools are great for that. What I think is less easy is when you have like, okay, we did this, we tried 3 different models, we didn't get good performance. Now we're going to go back, we're going to change the set of classes that we're looking at and change the magnification of the slides that we're looking at and the patch size. So the model changes, the data changes, like everything changes. So if you're in that phase of your project, then these tools I think are harder to use, unless I'm wrong. Maybe somebody can correct me if these are good for different sort of data parameters, like we're grabbing patches like this, using this policy, using this class set, then that would be great. And I would be happy to revisit that.
Exposing every parameter from patch extraction to training parameters to, inference and data set stuff. I mean, you're talking about a lot of stuff to record and keep track of. Just naming those and creating the interface for that is a significant amount of work.
Yeah. I think version control is a good option, because if you script out all of those things, then you could say, okay, we got these results with this Git hash. And so then you can check out exactly that set of code and run it again if you need to. But again, that requires discipline, right? You got to commit your code, make sure that it works, make sure that somebody else can use it, right? And if you're in a lab group with more than one person in it, then they're going to mess it up somehow.
I think the computer science programs should teach students to do this. So when they come to us, they already are, aware and know how to do all this. That's my my solution.
What's interesting is
what a life.
What's interesting as well is you may not know that Git also has pre-commit and post-commit hooks that you can write in Bash, Python, etc. So if your goal is to, for example, make sure that all of your code, if your requirement is that all of a person's code passes lint before it's committed, you can actually run that and reject the commit request if it doesn't run. And those scripts are highly modifiable. So if your goal is to say, I need to have an ID number to another software that's valid, you can write a small snippet of code that actually goes and parses that commit message to make sure that that ID actually exists. It's effort to set it up, and of course it takes discipline to actually do it. But once you kind of have that in place, and I've used this in a company where we had people committing code that didn't work, so we basically reject all commits unless a small subset of that we were using Java, the small subset of the JUnit tests passed, and then people committed a lot less, but everything that committed resulted in a build that actually compiled and that actually worked. And over the course of months, people naturally became better at not committing garbage because they knew that their garbage was going to be rejected anyway. So it's also kind of a long-term grind on their psychosis to make them better programmers. People hated us though. When we set it up, they hated us for like weeks. But I was like, you're basically telling me that you were trying to commit code that was going to break the build. Who should be angry at who here? And they go, oh, okay, well, maybe I didn't mean that. Okay, well.
Yeah, writing tests is something that I have a huge blind spot for. I don't ever do it. I should.
Super huge blind spot. Very little testing.
Yeah.
That was actually one of the questions I was going to ask at some point, but I'm thinking we're not going to get to that today. Sort of in the same line, do you have any like favorite or hidden gem packages you want to share with everyone else? Yeah, I already saw that a bunch of you guys posted stuff in the chat, but anything else that you came across recently maybe, or that you use regularly and you're like, everyone else should know this exists and it's like super useful?
I have a lot of productivity stuff. So in Windows, I use Greenshot, which is free and open source for taking screenshots, and it allows you to directly annotate those screenshots in high quality. You can save them in different ways. So I find that tool is utterly fantastic. I've started using a clipboard manager that's free and open source called CopyQ. So it at least keeps track of like an infinite number of things that I've copied into my clipboard, including images and other binary data, so that you can kind of pull it up lately and push it forward. Someone said Shapely. Yeah, so Shapely, I use a lot of Jupyter. There's a package called TQDM in Python that creates very nice progress bars that also estimate time built in. So that's, I think, pretty useful and easy to use as well.
For productivity, I like a tool called rclone. I have to deal with a lot of like cloud storage and moving data back and forth. And it's just a really nice way to do that from the command line. They have a Docker, and that's just saved me a tremendous amount of time.
Yeah, if you're doing computer vision, Albumentations is really useful. I found it to be very easy sort of drop-in replacement for PyTorch. And that's what I like to look for, is drop-in replacements for stuff, so that the cost of using it is less. That's why TQDM is really nice. I'm bad at it, so it kind of wreaks havoc on my log files, but if I'm looking at it in the console, it looks really nice. Because we do a lot of sort of server client stuff, any text editors or code editors that enable you to use SSH is really nice. So if you use Visual Studio Code, you can look into the SSH functionality of that. I have it so that it'll open up a project folder on a server, and I can edit the code as though it was a local directory, which is really nice. And then you can open up a terminal and you can run commands on the remote shell using that. So submitting Slurm jobs and so forth. For local development, I use Emacs because I'm a huge nerd and that's just what I do. But for that kind of thing, that's what I like.
So I do a lot of PyCharm as well. And what's interesting about PyCharm is that if you have an academic email address, you can request a professional license, which will allow you to do remote debugging on servers via SSH. So
Nice.
you can basically just point it at something that's running somewhere else and then debug it on your local laptop without any lag or any of that sort of stuff. And that's pretty helpful.
Did any of you guys ever like experiment with like QPy or the QKIM packages? There's like a bunch of like CUDA implementations of Python libraries.
Yeah, they work well. They're fast. It's still a moving target. Quite a few of us are involved in this Monai project in an advisory capacity. And, I think that the software that NVIDIA produces is excellent. I would definitely keep an eye on that.
Okay, so the next sort of question will be, since we're like 5 minutes to the end, more of a fluff question, I guess, which is going to be, since I guess maybe COVID restrictions will be less of a thing in 2022. What sort of conferences do you plan on attending, or do you recommend people to attend in 2022 or even the years beyond? And is there any other event you'd like to shout out, you'd like to recommend people to check out?
Everybody yell at your favorite conferences. We submit to SPIE and USCAP every year, which are conferences in the US. SPIE is medical imaging focused with a digital pathology sort of sub-conference. And then USCAP is the United States and Canadian Association of Pathology. That's the sort of clinical conference that we submit to. Has a lot of AI companies and AI projects that are starting to come up through that venue. PathLake, a lot of us on the panel sort of contribute to PathLake. The HEMA conference, Histological Image Analysis Conference. And I don't do it anymore, but I used to submit to MICAI and ISB conferences. I'm not, I'm saying the acronyms because I don't always remember what the acronyms stand for, but those are the big conferences that I usually go to.
I organize with some partners in pharma a workshop at ECML PKDD. It will be back in person this year in Grenoble, France. We're hoping to organize a mountain climbing outing. I like small meetings these days. I like to, go and interact with a smaller group. And so they're very interested in digital pathology. So if people are interested in submitting abstracts, we would look at those carefully.
I think for me, a question is always what audience we're trying to reach as well. A lot of our conference papers are really kind of advertisements for finding collaborators with external data sets. So we're more likely to go to, let's say, pathology or disease-specific conferences, in fact, to say, hey, we're researching this particular type of renal disease. Like we go to this American Society for Nephrology, say, is there anyone else in the audience that would like to be a collaborator so we can have an external validation set and that sort of thing. And I think that's a lot less likely to happen at digital pathology conferences, which are more focused on presenting results and sharing technology versus kind of the clinical applications. So I think there's kind of a two-pronged approach that can be taken there.
Does anyone go to any programming conferences? Like I know that Python has a bunch of national conferences in the US. Does anyone go to any sort of computer engineering style conferences?
We've sent people to PyData before. I think that's a really nice one. If you like programming, you get to really see like all of the cool new packages. I would recommend that.
I think it's ultimately, and maybe this is something that we can do, is think about creating a mailing list for kind of sharing tools and that sort of thing. So I think the original inception of this conference was Lee and I were talking, and we realized that we use completely different tools for a lot of the things that I was surprised by. And it's ultimately, especially working remotely, but there's usually not a lot of digital pathology people in the same place. So you have like a bunch of different oncologists working together, and they kind of can share information, and a bunch of pathologists working together and they share information. But I think it's pretty rare that you have, multiple digital pathology professors all in the same university. Maybe the Netherlands folks are a little different. But I think most of us are pretty isolated, and it becomes difficult to figure out these small little nuanced things that come out in the ecosystem.
And even this discussion, I mean, because it's the first one, we sort of focused on a lot of big topics and broad topics, but like I don't know how much of a broad interest it is, but I'm interested in seeing how people write their code and what the process is, you know, and sort of detailed stuff like that. Actually, you know, things like, does anyone use anything other than Python? Does anyone use like Julia or Rust or something? That would be kind of interesting to bring folks in who have experience in that.
No, we're keeping the Rust folks out. We have to set reasonable constraints here. You know, we were kind of like homogenizing on Python and PyTorch, and now, oh, let's just open this back up again. Oh,
yeah,
what kind of bizarre programming languages can we use?
Rust is not a bizarre program. So actually, there is an open slide crate for Rust, which I tried, and I was able to create a single patch using Rust. I was very proud of myself because I don't know any Rust. And so
3 days later. 3 days later, Scott.
I created one patch, and I was
imagine what you could have done with that time.
And that was 8 months ago. So
that's quite funny.
I think a lot of bioinformatics stuff is still in R, so we need to keep that in mind as well.
Yeah.
Terrible language, by the way.
Sweet. So I think that's it, right? That's the time we have today.
I put up a poll to just kind of gauge everyone's you know, thoughts on the meeting. Unfortunately, there was no text box available, so I was going to ask for comments, but that appears to not be an option. If you have additional comments or thoughts or suggested improvements, I think you have probably one, if not all, of the people on the panel's email address. Feel free to reach out to us.
Tweet at Andrew if you have any.
Yeah, definitely tweet at me. It's Andrew at notontwitter.com, or is it hashtag? I'm not really sure how the technology. You know, hashtag have a good day.
More of a curmudgeon than I am.
No, it started with a C. I was expecting a K. Learn something new every day. I think that's it, folks. So thank you, everyone, and have a good day.
Thanks, everyone.